let's get started. We'll slowly ease into things for anyone who's straggling or having trouble connecting. But hello and welcome to this session all about SCA6 research. So my name is Celeste Seward. I'm the National Ataxia Foundation's new patient engagement manager. So my job focuses on helping ataxia patients and family members get more involved with research and helping researchers get more involved with the ataxia patient community. A couple of housekeeping items before we jump into our wonderful talk today. If you need closed captions, you can turn them on using the button that has CC at the bottom of your screen in Zoom, and you can toggle those on and off if you would like captions. Please feel free to use the chat feature to connect with other attendees like you're doing now, letting people know where you're from and where you're joining from. But if you have any questions specifically about the talk today that you'd like to chat about, if you could please use the Q&A feature. So that will send them directly to us. So in the question period that we'll be able to see them and respond to them. So if you're chatting with other participants who are watching, please feel free to use the chat. If you'd like to ask a question specifically, just drop that in the Q&A window, which you can find at the bottom of your screen as well. And you can put in questions into that Q&A window at any time during the presentation, and then we'll get to all of them at the end. So with that, those are our housekeeping items. Let me give you a little bit of an introduction to our speaker today. So Dr. Allana Watt is an associate professor and principal investigator at McGill University. She completed her PhD at Brandeis University in Massachusetts in the Turrigiano lab, studying homeostatic synaptic plasticity, which is quite the mouthful. She then moved to the University College London in the United Kingdom, where she studied cerebellar development at the Häusser lab. And Dr. Watt started her own lab at McGill University in Canada in 2011, focusing on cerebellar development and plasticity in both healthy brains and in animal models of disease. So thank you so much, Dr. Watt, for joining us today to chat about SCA6 research. The floor is yours.
Thank you so much, Celeste. You can hear me all right?
Yes, we can.
Good. So I have to just say that I have a little bit of a cold, so hopefully I won't be sneezing too much, but I apologize in advance if I am. I'm going to show you some slides, and I'm really excited to get this opportunity to talk to you today about the research that we do in my lab. And first, I want to talk just a little bit. That was sort of the introduction, the very nice introduction that Celeste gave. And I'm just going to, sorry, I'm just adjusting things on my computer. I just wanted to point out, for those of you who aren't all that aware of what research is, because it can be a little bit confusing. Most of you probably interact with doctors like MDs, and that's not the kind of doctorate I have. I have a PhD. So what does it mean to have a PhD? Well, this is the little cartoon showing you this. This is I'm just going to turn on my laser pointer. So, hey, my pup is sick, says the mouse. Are you a real doctor or just a PhD? So that's right. I'm a PhD, which means I'm not a medical doctor. I'm the kind of doctor that does research and doesn't work with people. So you probably haven't maybe met too many people like me in your journey with SCA6 so far. Okay, so I have a little video here that I usually talk about when I talk about ataxia, and I know you probably don't need to give this introduction, but I just wanted to talk about the deficits that are associated with ataxia, and then the brain region, which is the cerebellum. You might know that the cerebellum is affected. This is the cerebellum actually means little brain, which is kind of tucked up at the back of the bigger brain, but you might not have seen this. This is actually some of the evidence showing that the SCA6, the brain, the cerebellum is affected. So this is just showing you the size of the cerebellum overall is changed. And then actually Purkinje cells, which we're going to talk about more, are actually dying in patients with this disease. So how this is showing you data from patients, which is something that sometimes people do look at. But what is this disease caused by, you probably know this already, that this is caused by expansion of a polyglutamate region in a gene. And the gene that we are looking at, I'm going to call it the SCA6 gene, subsequent from this, but I wanted to explain a little bit to you. It's called the CACNA1A gene. I know that's not super informative. This is this kind of name shows us that it was named after a channel. So it's thought to have it's thought to encode a subunit of a channel, and this is something that's actually enriched in Purkinje cells. So these PQ channels. The P in PQ channel stands for Purkinje. So these Purkinje cells have a lot of this channel. Work from Chris Gomez's lab, another researcher who also works on this disease, has also shown that there's another product of this gene. So there's some genes that can encode more than one protein. And so he's showing that this is encoding transcription factor. And that's work from his lab almost 10 years ago, but it's been very influential. And right at the end of my talk, we're going to come back and talk about this and what does it mean to be a transcription factor. So as I'm sure you know, that this is a progressive disease. It comes on at some point in midlife, and then it kind of worsens. And we know that there's some cerebellar loss and Purkinje cell loss, especially at the end of the disease. So we don't work on patient tissue. We don't work on cell lines. What we do is that we have work with a mouse that kind of recapitulates this disease. So this mouse has also a polyglutamine expansion. It's a much larger expansion, a much longer expansion than what people have. And when this mouse was created, what happens is that it kind of recapitulates, we like to say, the disease in a mouse. So we have motor deficits that come on kind of in the middle of the mouse's life. You probably don't know how long a mouse lives. About 2 years is about how long a mouse typically lives, and 2 years is pretty long. They're pretty old for a mouse. So 7 months is kind of the prime of the mouse's life. This is around when we start to see motor deficits, and those motor deficits worsen. And I'm going to show you how we study this in a mouse, like how do we study ataxia in a mouse? And I'll show you that in a little while. And then we've also been able to show that there is also Purkinje cell loss in this mouse, but it's actually very late. We don't see it over here at the beginning of the disease. We see it very, very late at the end of the disease, which is kind of encouraging because it suggests that there's some long period of time before Purkinje cells die, when maybe intervention might be even easier. So, as I said, what has been done in this particular mouse is that they've taken this gene and they've inserted these polyglutamine tract, this extra long polyglutamine tract. This was actually done by another lab. This was created by Huda Zogby's lab, and the researcher who worked on this was Watazi, who now also has a lab of his own in Japan. And so this is the mouse that we use. So scientists like to share most of the time. And so they've made this mouse available so that other people can study it too. Another mouse is also in use, and it's a little bit different. They've only taken that sort of a little fragment of the mutated gene and they've kind of put that in extra. So then the mouse has kind of the regular gene and also this extra copy of this little fragment. And that also can tell us, we hope, something about the disease and how it develops. And that is work from Melanie Mark's lab in Germany. And that's, I'm talking work about a paper that she published in 2015. Okay, so most of what I'm going to be telling you about is the work that's been going on in my lab. And I'm just showing you pictures of the two people who have been working on this the most. um, um, or who I'm going to be showing you their work. So the first is this Dr. Shriram Jayabal. So he was my very first PhD student, actually, and he then graduated and is now doing a postdoc, sort of further work in another lab. And then Anna Cook, who is a PhD student, a current PhD student in my lab, and she is very close to finishing and graduating and getting her doctorate. And she's been working on this project for several years. So I just wanted to tell you a little bit more about why, what are the advantages of using transgenic mice as a model system? So I think actually, personally, sometimes when you make a mouse model of a disease, it kind of looks like the human disease, but sometimes it doesn't. I would say that this model is a very good model because it really seems to kind of recapitulate some of the changes that we see. And just to be clear, there's some really weird things that we do in science. And one of the things is that we call the control mice, so the mice that have the regular gene, we call them wild type. And I'm going to abbreviate that in places as WT. And so I wanted to make sure that was clear because I remember when I first became a scientist, I'm like, wild type, what does that mean? It just means like without the disease, so the kind of control. And so one of the advantages of mice is that we're using what's called an inbred mouse strain. And so really, and we use like siblings, so they're litter match controls. So mice have big kind of a litter of maybe 6 to 8 pups. And so we can use the wild type mice and this costs 6 mice, and only the genes should be different. They're very, very similar genetically. So that kind of reduces variability, allowing us to hope that changes that we see are really due to changes in that gene and not something else. So we're very careful about the way we do our research so that we have like the right controls. Okay. That's just to kind of explain a little bit what we're doing. I also wanted to point out that this isn't the only way that people study these diseases. Sometimes they might use like a cell line, and that's just sort of a bunch of cells growing in a dish. This is something, I took this picture. These are called hex cells. It doesn't really matter, but you can express particular genes in these cells very easily and then see sort of what kind of changes at the cellular level. And that's something that Chris Gomez has done, for instance. He used hex cells in his 2013 paper. So there are multiple ways that researchers can can pursue these questions depending on what kind of question we're asking. In my lab, we really work on mouse models, and that's really kind of the kinds of questions that we're asking, because we really are trying to understand like what's changing in the brain of the mice when they start to have disease onset and as things worsen as well. So let me explain. Okay, so why are we focusing so much on Purkinje cells? So on the one hand, it's because these are the cells that are going to end up dying. So they seem to be very vulnerable. So it's kind of a good idea that maybe to start by looking at those cells. This is like a little schematic, and there's a whole bunch of different cells here and kind of like arrows coming in and then going out. But one thing to kind of understand is that these cells are kind of a bottleneck. So they're the only thing that's going out of the cerebellar cortex. So if there's a change, something to do with them, that's really going to be signaled to like that sort of, they're kind of carrying the signal of the cerebellum, and therefore, if there's a change, that change is going to be reflected in the output of the cerebellum. They are the output of the cerebellum, basically, and that's what this one kind of line going here. There's only one arrow going out, and that is the Purkinje cells. They're kind of the way the information flows out of the cerebellum. Okay, but I'm going to just point out something that I think is kind of interesting, And it's not something that I have a particular answer for at this point, but it is one of those things that I think is kind of interesting and gives us something more to think about. And that is that there are a lot of cells in the brain, not just Purkinje cells, that express the SCA6 or the SCA6 gene, the CACNA1A gene. I'm just going to call it the SCA6 gene. But it seems, so we know that Purkinje cells have a lot of this gene, but they're not the only ones. But they seem to be the only ones that die, and we don't really understand why that is. So that's one of those big open questions that we're hoping we can try to address using our mouse models. Okay, so there's something else I want to tell you about Purkinje cells, and that is that they fire spontaneously. So, excuse me, what you can see here, I'm actually going to show you guys some electrophysiology. And this is kind of a little bit crazy, but I think it's actually really interesting to see. It doesn't really matter exactly what this is. What I'm showing you is kind of the electrical properties of cells, of neurons. And so neurons actually fire these signals, which we call action potentials or spikes. It kind of makes sense that they look like spikes, I guess. And they fire really regularly, and they fire in the absence of any kind of information coming in from the outside. So the way I think of this, this regularity and this kind of constant firing, it's almost like the Purkinje cell is a pacemaker in that it's kind of keeping the beat relatively well. Okay, so there are several ataxia mouse models where we see decreases in the firing rate, and also some where we see decreased firing regularity. And one of those is SCA6. So when we look in our mice at a time when what we call disease onset in a mouse, when we're just starting to see ataxic symptoms appear, we see that there are changes in the Purkinje cell electrical properties. So this is just showing you how we actually do these experiments. We bring an electrode up to the cell and we kind of listen in on what the cell is doing. And so in wild type, you can see that they're firing this, they're going down, but it doesn't matter at all. You can still see these spikes, these action potentials. In our SCA6 mice, we see that the action potential rate has gone down, so there are fewer of them. And they're also a bit more irregular. So this kind of pacemaking property that I told you about for Purkinje cells seems to be disrupted. And that's something that we think is actually quite important. So I'm going to show you a pretty busy slide now, and it's a little overwhelming. I'm going to talk you through it. Don't get too overwhelmed. This is the kind of stuff we make. This is from a review paper that we wrote in my lab. And this was trying to kind of explain a lot of things. So basically what I'm trying to show you is over here is like a healthy Purkinje cell. This is also what Purkinje cells look like before symptoms onset in SCA6. But then depending on a whole bunch of different things, so it could be different kinds of diseases, channel diseases, developmental diseases, things like that, a whole range of diseases, you end up having Purkinje cells which fire differently, typically with low rates. And this isn't just seen in SCA6, this is seen in SCA1 and SCA2 and I think SCA3. And pretty much every mouse model of ataxia where we've looked for this, we've seen these kinds of changes. It's also something that's been shown in Huntington disease and also most models of autism. So this is sort of different, a different population of Purkinje cells, but they are kind of having, this is also kind of early in development. So these are kind of showing here, these mice down here showing kind of deficits that are like motor, and then there are also kind of cognitive or social things, and that would be for the autism models. Interestingly, we've been able to show, and I'm going to show you some of that data now, and others have shown this too, that if you do something to Purkinje cells that can rescue this regular firing to make it fire at higher frequencies, that this is associated with at least some sort of rescue in at least kind of partial rescue in kind of motor and also cognitive behaviors. So we think that this is pretty, this is pretty good evidence that the firing deficits are actually contributing to these motor things. And because when we change the firing, we can actually improve them. Okay, so why would that be the case? So now I'm going to just try to show you in a nice analogy. So Purkinje cells, as I say, they can fire at high frequencies. Some of them fire at high frequencies, some of them fire at low frequencies. You kind of get a wide range. And I like to think of it something like this. If we think of it from the whole brain perspective, let's imagine that what the cerebellum is doing is giving us some sort of output that would be like a piece of music that an orchestra is playing. And so I'm showing you here a picture of the Montreal Symphony Orchestra because I'm based in Montreal and we have a beautiful symphony orchestra here. Now let's imagine, so each person here is a Purkinje cell and they're all tuned to their different frequencies. Now let's imagine that we take out all the people who can play the high frequencies. They're just gone from the orchestra, but the orchestra is going to try to continue to play without those high frequencies. Well, then the output, the music is not going to sound the same. And we think this is sort of an idea for why what seems like perhaps a small change in Purkinje cell output is nonetheless having a relatively big effect, at least on our mice. Okay, so we got started on this project that I'm going to tell you about, actually from looking at a paper from a patient paper. And so this is something, we read a lot. We call it the literature. So all the papers that have been published about different forms of ataxia, in particular SCA6. And we think about, how can our research get inspired by this? And so this was showing that there was a change in something called BDNF. And so I'm going to tell you a little bit about BDNF here. BDNF was low in the brains of patients with SCA6. And so what is BDNF? It stands for brain-derived neurotrophic factor, not the catchiest name. It's a BDNF binds surface receptors called TrkB and signals. I'm going to show you a little bit of data about this in a minute, so you can you'll see kind of variations of this. And it's considered a growth factor. Like in development, it's really, really important that we have BDNF or the brain doesn't develop normally. But it seems to be important in the adult brain as well. Although to be honest, there hasn't been that much studied about how BDNF works in the adult cerebellum. I was surprised, but there you go. Sometimes people don't study everything in every brain region. And there's certainly been more attention focused on other brain regions. So this is just a kind of simple analogy, but BDNF is considered, a little bit like a fertilizer for the brain, where it just seems to be enhancing brain function. So here's a plant grown with fertilizer and a plant grown without fertilizer. You can see there's a big difference. And that's kind of like what we think BDNF is doing. So we thought it was really interesting that this had been reported because it was surprising to us, and we didn't actually know what BDNF was doing in the adult brain. So we wanted to see if that was true also in our mice. And so we looked at a couple of different ages, and I'm just going to show you 7 months of age, which is kind of our disease onset in our mice. Excuse me. And so what I'm showing you, we looked at this in several different ways, but I wanted to show you some pretty pictures. It can be really nice to see. So what we've done here is we've taken the cerebellum and we've been able to use antibodies to label different proteins. And so here's one that's kind of a marker for Purkinje cells. And you can see that the Purkinje cells look really nice in our control mice or wild type mice and also in our SCA6 mice. If we look at the BDNF levels, yeah, they do look reduced in our SCA6 mice compared to controls. And this is just showing you that other things don't seem to be affected. So it really seems to be limited. And you can kind of see the difference of colors here. This is a little bit more orangey than this down here, meaning that there's less yellow in the picture. And then we quantified that and we could see that there was indeed a big reduction. Okay, that's not totally surprising because the BDNF has been shown to be reduced in other diseases. I'm showing you some data from Alzheimer's disease. It doesn't really matter that much. This is actually showing patients. And so in Alzheimer's disease, it's a different brain region that's affected. It's a hippocampus as well as other brain regions. And so BDNF is low there, both in patients and also in mouse models. And so that's kind of interesting as well. It's also been shown to be reduced in other ataxia. So it's not like this is the only disease where this is seen. That's actually kind of a good thing, as you'll see, because people have more tools to study BDNF because of that. So this is showing you data from a mouse model of SCA1, where it's also been shown to be reduced. So can we increase BDNF levels in the cerebellum, or sorry, in the brain? Actually, we didn't really know if this was possible in the cerebellum, but it's really well known that you can increase BDNF in the brain, and you might know about this research yourself because it's kind of well known, and that is by exercise. So exercise is one of those things that has been shown to increase BDNF in the brain. This is an old study just showing you that. Here's a brain slice from a sedentary rat. This again is a hippocampus and cortex, and then an exercise rat, and you can see that that BDNF levels do in fact go up. Okay, so we wondered if we could do the same thing in our mice. We gave them running wheels, and mice love running wheels. Like, you just cannot believe how much they ran, like for hours and hours and hours a day. Okay, so they do run. Our SCA6 mice run, and so we either give them a sedentary mouse gets a locked wheel that can't rotate, and then an exercise mouse gets a wheel that does rotate. And then we're going to do a behavioral assay. So you'll see how we're looking at whether or not there's any kind of ataxia in these mice. And you can see that on subsequent weeks, they kind of run more and more. Our diseased mice are running less than our wild type mice, but they're still running a lot. Okay. So what happens to BDNF? So it's pretty cool, but exercise, you can see here this yellow panel, exercise seems to at least partially restore the BDNF levels in the cerebellum. So just like we saw in the hippocampus, exercise seems to be able to restore BDNF levels in the brain. And then we wondered whether or not it was going to affect ataxia. And we use something called a rotarod to look at this. So the rotarod, I'm showing you here. We put our little mice on this rod and they try to stay on it. And it's kind of a, a skilled task. Like they don't love it, but but they prefer to stay on to falling off. And so what happens is our mice, the SCA6 mice, they fall off more than our wild type mice do. And that's what I'm going to show you here. So this is showing you just several days. They kind of get better with time. So they learn to stay on the mice. This is on the rotarod. This is the latency to fall. So they want to stay on. And our wild type mice stay on for longer. Our SCA6 mice, they don't do as well, right? They're learning a little bit, but not that much. And there's a big difference here. Well, what about with exercise? So we did the appropriate controls where we gave our exercise to the wild type mice, and we didn't see any statistically different. They're a little bit better, but not significantly better. They're kind of overlapping. But when we gave the SCA6 mice exercise, we actually saw that there's a partial reversal of ataxia. So they're doing kind of in between the mice without exercise wheels and our wild type mice. So that's pretty encouraging. So what about Purkinje cells? I told you so much about Purkinje cells. I gave you a little lesson in the cerebellum and how cells act. What about Purkinje cells? I'm sure you're wondering that question. And it turns out that the Purkinje cell firing deficits are restored by exercise. So you can see this here. The SCA6 exercise mice are firing at higher frequencies than they are with the sedentary mice. So that's also pretty exciting. And that's what, sorry, these little stars up here are just telling us what's different from each other. Okay. So just to summarize this part, we see that BDNF levels are low in the cerebellum and exercise can increase them. It can also reverse ataxia, at least partially, and also reduces the Purkinje cell firing rate deficits. Again, sort of supporting this idea that if we can make Purkinje cells fire better, fire more like wild type cells, then they are going to lead to a behavioral improvement. And, you know, this is not maybe completely surprising because exercise is one of the only things that's usually recommended for patients, as perhaps you know, and this is just showing you some papers that show that. However, so we had a kind of something that, you know, mice are not the same as people. And whereas you can say to a person, exercise is good for you, you should try to do that. And often, but not always, the people will listen. But that's not the way it works with mice. We can reason all we want with them, but they're not going to listen to us. And what it turned out was that when we gave mice later, after their ataxia had already sort of started and we gave them running wheels, it didn't lead to any significant improvement in their behavior. However, they basically weren't exercising very much at all. The amount of exercise that they had was very, very low. We tried it in a couple of different ways and we weren't able to, you know, reason with the mice and get them to exercise. They just didn't want to for whatever reason. So we wondered whether or not we could modulate BDNF levels in other ways. And it turns out, this is just taken from a review paper, that like other people have thought about this because BDNF is known to be so important in other diseases such as Alzheimer's disease. There's already been some progress made with these sorts of things. So there's, you know, you can increase BDNF by physical exercise, cognitive stimulation, perhaps. I mean, we don't know if that's true in the cerebellum. Other ways, gene delivery, kind of molecules that like mimic BDNF. There's a whole bunch of different approaches that people have been thinking to, like, how can we make BDNF higher in the brain? What I'm going to show you is one idea based on these BDNF mimetics. I think some of the other ways are really promising too, but maybe they're just a little farther away. So we wondered if we could mimic BDNF, and in fact, there are drugs out there that do such things, and one of them is called DHF, or 7,8-dihydroxyflavone. And this is something that is bioavailable orally, which means that you can take it orally and it'll get into your bloodstream and get into your brain because it crosses the blood-brain barrier. And so it should act on the same receptors that BDNF typically does. It has also been used in mouse models of Alzheimer's. So we gave mice DHF for a month, and then we saw that there was an improvement. So this is just our SCA6 mice and the SCA6 mice with DHF. You can see that they're doing better than the mice without DHF. And so we think that that's showing us that DHF is acting very similar to exercise. They also see a change in the firing rate, just like we saw with exercise. We see that frequency of Purkinje cells firing goes back up, very encouraging. As we know, this is a progressive disease, so we wondered whether or not we could give DHF for a longer period of time and we could see benefits. And so this is something that we tried. We did this for a couple of months. This was actually work we did during the pandemic, and we ended up having to, we had to kind of curtail these experiments. But what you can see is that with DHF, we see a kind of improvement that seems to last for several months. So to summarize this part, we think that BDNF is, DHF is mimicking BDNF and mimicking the effects of exercise. So it's acting like the BDNF does in our brain, and that makes it a promising therapeutic. And I've been talking to different clinicians about, whether or not this is something we could do, pursue clinical trials for. The clinical trials is not something that I have any experience with. As you understand probably now, I work with mice and study things at the kind of cellular level, but we're really hopeful that someone is going to be interested and want to do this. We're also looking for new approaches to find changes in the brain in SCA6 mice. So, excuse me. just to give you a little kind of taste of that kind of work, remember that there was a transcription factor that was altered in our mice. This is work from Chris Gomez. So we got kind of inspired by that. Because if there's a transcription factor, so you might not know what a transcription factor means, but this means that it controls the expression of other genes. So we know that there's this one gene mutated, but if it's controlling the expression of other genes, there may be hundreds of other genes that are changing in the brain. And so we looked at that doing something called bioinformatics. And this is something that has been done by a fantastic grad student in my lab, Sophia Leung. And so she looked at wild-type mice and SCA6 mice, and she took the cerebellum, and she looked at the expression of different genes. And so this is fairly technically challenging for us. But what she found was that out of about the 25,000 genes that are expressed by mice, about 500 are differentially expressed, with about half of them being upregulated and half of them being downregulated in SCA6 mice. So it does suggest that there's a lot of dysregulated genes. And now we're trying to follow that up and try to understand more. Some of the genes that we are looking at, we had no reason to think that they would be involved in SCA6, and so we're really interested in pursuing that further and maybe finding new things that could be influencing disease. Okay, so I just want to acknowledge all the people in my lab who make this work happen. I think I mentioned Anna, Shriram Jaybal. We have some Sophia as well, and then some undergrads who helped out, like Jackie and Genevieve. I also want to acknowledge my funding sources and collaborators, and I'm happy to answer questions at this point. So thank you very much for coming and listening to my talk. I'd love to hear from you.
And thank you so much for giving this talk. Yeah, if you want to, if you want to keep your slides up, you can. If you'd like to take them off, that's totally fine as well. Either works. We have some questions that are starting to roll in. Just a reminder, if you do want to ask Dr. Watt a question, there's a button at the bottom of your screen. It looks like two little speech bubbles. It has Q&A on it. If you click that, you can type in a question and it will be sent to me to read out. And we have the first few coming in. So the first is a bit of a clarification question. So you talked about BDNF, but you also talked about HDF. So why would you use DHs? My apologies. So why would you use something different? Is it possible to just supplement BDNF directly?
Yeah, that's a great question. As always, the answer is it's a little bit more complicated than what I told you. So BDNF, I told you about it acting on this receptor called TrkB, which is, I didn't talk too much about that. But BDNF actually has another receptor, and that receptor is, it's thought to be kind of have less affinity for BDNF, but if you were to just add a whole bunch of BDNF to the brain, that can actually often be bad for the brain. Like so the way we dose it, like if we just kind of add BDNF to the brain, doesn't seem to be effective. So it's a really good idea. Why am I going to this complicated thing of using DHF when why not just use BDNF? But BDNF on its own, it's thought to be through its actions on this other receptor, can lead to, that receptor is actually sometimes called the death receptor. So it seems like really, really high levels of BDNF can lead to cell death.
Oh, okay.
But DHF doesn't activate that receptor. So that seems to be, why DHF is probably better. So if we could,
you're able to get the benefits, but not the potential negatives.
Yeah, if we could dose it perfectly, that wouldn't be an issue, but I think we just can't at this point. Yeah,
oftentimes it's a Goldilocks situation where too little of something is not good, too much of something, you need it to be just right in the middle.
Exactly.
So we have our next question here, also about BDNF. So you talked about how exercise can influence BDNF levels. Can BDNF be increased by any kind of diet, like restriction or supplements, to your knowledge?
Yeah, I think I include, oh, I'm going the wrong way. I think I included this slide. So there
Mhm.
is some sort of idea that diet restriction, this is this is not something I know that much about, and I would
Of
take this
course.
with a big note of caution. So when people have looked at BDNF in the brain, they've often done it with things like mice, and they maybe have just looked at specific regions.
Mhm.
So what I would say is that it's not clear that that's going to be the case in the cerebellum as well, but there is some evidence, I think, that could be the case. So one thing that I didn't talk about, but I think is really interesting.
Mhm.
Sorry. Um, I want to just go back
Of
here
course.
to our BDNF levels, right?
Mhm.
And if we just look at our BDNF levels here, it maybe isn't kind of obvious, but exercise on its own didn't lead to an increase in BDNF in our wild type mice. So that
Okay.
kind of suggests that BDNF is at really like kind of saturating levels normally in the brain,
Mhm.
and that this only, this kind of rescue is only happening when the BDNF levels are lower.
Mm.
And so we really don't know too much about like how BDNF is normally kind of working in the cerebellum in particular. So I think
Mm.
that this is maybe, it's something that actually we're interested in looking at more, like just to kind of understand how BDNF is being trafficked normally in the
Mhm.
brain, because we don't know too much about that actually.
Of
And
course.
sorry, when I say the brain, I mean the cerebellum in
Mhm.
particular. It's just been kind of understudied for some reason.
Mhm. And then we have a number of people asking a similar question. So I'll pick the phrasing that one person suggested. Could this
Okay.
be something that would only help SCA6, or could other kinds of ataxia be helped out from BDNF increases by exercise or by other means? To your
Right.
knowledge.
That's a great question. And I would say, the fact that this a reduction in BDNF has been seen in SCA1 is kind of it to me that really suggests that maybe this is a more general thing,
Mhm.
and maybe it could at least be used for SCA1, but perhaps other diseases as well. Like, so the reason we only know about one other disease is because there's only, kind of one researcher looking at this and that disease, and there just hasn't been that much studied. But to me, it
Mm.
would suggest, I guess, somehow it's like, well, if it's true for two diseases and we've only
Yeah.
looked at these two diseases, it's a pretty good chance. Yeah,
So we need to band together some researchers looking at other types of ataxia to see if it works in
exactly.
their animal models as well. That's very helpful to know. We also have a few people who are uh, interested in, well, they're asking if DHF can be prescribed right now, but perhaps you can talk about, because right now it's not available as a prescription. It's still being tested out to make sure it'd be safe in humans. But perhaps you can talk a little bit about how, if you partnered with a clinician, what would be the next steps to take the results that you have in mice that are really promising, and what would need to be done before this could be taken by humans?
That's a really good question. And so I think what we, I I think personally mice are good models
Mhm.
for disease, but they're not going to capture kind of everything about the disease, obviously. So the fact that it's working, it's promising in mice is very exciting. Um, I have talked to a clinician actually in the UK about this work, and it would be really, she's been interested, so there are things we could do. We could look at whether or not exercise works to increase BDNF in the bloodstream. We could
Mhm.
also look at whether DHF is also having some sort of effect. And so this would be, it's really important to do this in a clinical, in a real clinical trial where people would be either getting DHF or not getting DHF. And when I say real clinical trial, I mean like a double-blind clinical trial where you,
With
the
all the
participant,
controls.
yeah, don't know which group you're in, so that we can really see if there's an effect. Because I think, sometimes it happens that something we see that's really promising in mice for whatever reason, just does not translate into the clinic. And so it's not there yet, but I'm certainly hopeful that we're going to be able to find that partnership and find a way to move forward with that.
Yeah. So early days, promising, but still needs some work done in humans to make sure that things are safe and answer questions about how much DHF that a human should take, because humans are much bigger than mice. So still some questions left unanswered, but promising so far.
Absolutely. Yeah.
And then we have a question about the exercise that the mice did. So how, if you could chat with us a little bit again about how much exercise led to benefit, and how would you quantify or translate how much exercise a mouse does to try to figure out how much exercise a human should be doing, if that's possible?
Yeah, it's such a great question. I wanted to show you this. Here we go. So this is like how much each mouse ran over a month. And so, and compared to how well they did on their Rotarod score. And so you might not be used to looking at data like this so much, but this is like a really good positive correlation. Like most of the mice who ran very little did poorly, and most of the mice that ran a lot, or the few that ran a lot, are over here up at the higher level. So this is what we call a positive correlation. But look at these numbers. I mean, this is actually
that's a lot.
Bonkers.
That's very far to run. That mouse is getting a
kilometers.
very good workout.
Mice are really little. Mice, sometimes I despair when I think about this. I just think, how can they run like this? But mice, when you give them a running wheel, they run like most of their waking day, which is actually they're nocturnal, so that means like most of the evening. So they're in a room where the lights turn off at 7 p.m. and then they go on at 7 a.m. and they run for most of that time. That's just not how it is for people. We don't run like that.
We work slightly differently.
Yeah, exactly. But there has been research done about how to elevate, some research done about how to best elevate BDNF in the brain with exercise. And one study I know has shown that high intensity interval training, which is a very popular kind of, often in exercise classes and things like that has been shown to be good at elevating BDNF. But I would imagine that most forms of exercise and kind of the amount of exercise that you would feel is a reasonable amount, not like hours and hours a day, would be good at elevating BDNF. And so that's kind of like when people look at those things, they're looking at how much BDNF is in your bloodstream, and the idea being that would correlate with what's in the brain. It's not a perfect correlation necessarily, but
it's a place to start.
Exactly.
And then questions are rolling in, so we're going to get to as many as we can in the next 10 minutes. So another one we have is, so with exercise having a positive impact on BDNF levels, could exercise mimetics or other drugs that are connected to exercise like metformin be an alternative? Would you see similar benefits if you tested that in a mouse?
Oh, that's a really good question. Metformin is something, I mean, we just, gosh, I'm trying to think of how it's signaling quite differently than BDNF, although it's possible that their signaling pathways intersect. A lot do, but I don't know if we can make that kind of leap. I think there's maybe
totally
like
fair.
one or two steps that I'm not sure about, but it would be interesting for us to test for sure. Metformin is an exciting, there's certainly been some promise with that in different diseases.
Then we have another question here about the SCA6 gene and protein. So does it matter if the SCA6 protein is firing more than it's supposed to or less than it's supposed to? Or does that differ between SCA6 patients?
Yeah, so as far as we've seen, it's overall we see a reduction in firing. We've never seen an enhancement of firing of these Purkinje cells. It's possible that that would, I mean, you could argue that would also lead to dysfunction, this kind of Goldilocks idea that sort of, you have to be in that just right zone. I think it's typically not It hasn't necessarily been observed though, and that might just be sort of from a kind of the biology of the cells. It's like it's more likely to things when if things aren't working just right, that the firing is going to go down because it's actually like the firing that we're seeing. I was showing you something like ranging from 10 hertz to 200 hertz. 200 hertz is actually like 200 action potentials per second. It's
That's a
kind of
lot.
flabbergasting that your brain could kind of be doing this. It's a lot, and it's presumably quite energetically like intensive for a cell to do that. So it's more likely, I think, for it to kind of go down if it's not functioning just right. Yeah.
Fantastic. And then I'm just skimming through some of the questions so I can figure out which one to ask next.
Sure.
You have fantastic questions, folks. I am overwhelmed by the awesomeness. And then someone's asking if you could talk about, again, the difference between the impacts you saw in the mouse when they started exercise early versus when they exercised late in terms of when they started to first get symptoms. And do you
Right.
think that if a human started exercising earlier before they started experiencing symptoms, would that delay things maybe in terms of when they would first start getting symptoms?
Yeah, that's an interesting question and something we would like to look into more, because what we saw was that there was sort of a reduction in the symptoms. And does that mean that there's actually a delay, or that the symptoms are just coming on a little bit lower? It's actually really hard for us to know that kind of thing in a mouse, because we tend to have to kind of like, we do many mice. Maybe we have 10 mice that we look at, and then we kind of have to average them, which is so it's a little bit hard, but it would suggest maybe that there could be some sort of delay, or just that the progression goes more slowly. Those are things. Yeah. I mean, I'm hoping I've just written a grant to like address some of those questions, because I think it's a really important to understand in more detail. Yeah. So I don't think I really could answer. Both seem
That's
possible.
fair.
Yeah.
And then we have another question about the mouse model you're using. How do you know that the SCA6 protein is being expressed in the cells that you're testing and the mice that you're testing? And could you talk a little bit more about that?
Yeah, that's a great question. So how do we know? Yeah, we sometimes have looked at the protein expression, like just like I showed. We can look at the protein expression like this, where we actually look at like where it is in the tissue. We also like routinely we would sequence these mice to make sure that they still have this mutation, because we just want to make sure that our mouse model is in fact still kind of doing. So each time we have a litter of mice, we take a little tiny bit of tissue from the mice, and then we do PCR, which is just the same kind of thing that we've done like with COVID for all this time,
to
where
test to
we
see if you have it.
yeah, exactly, to detect to see if we have it, and we can we have a predicted like shift in a little band of DNA, and that's what we do on a weekly basis. So this is kind of like lab housekeeping things. I hire some students who just this is sort of their job basically, is just to do this, and then they get to have a little experience working in a lab and see if they like it, and we get some of this kind of housekeeping done. But then every, I think, 6 to 9 months, we kind of also would send this to a company that would actually sequence the gene and make sure that it's still there and that there's no difference. Yeah. It's
That's
a really
helpful.
important thing.
Yeah, just to make sure that the mice that have SCA6 in fact have SCA6.
Exactly.
Then we have
We're always
another.
worried.
No, exactly. You want to make sure that everything's good, so the results you're getting are real and reproducible.
Absolutely. Yeah.
Then we have another question here. Could you chat a little bit more about what cell firing means for neurons, and what does it do in the brain?
Yeah, that's a great question. Yes. Where was I going? So if we come here to this sort of like little schematic, and you can see that there's like cells, there's sort of an input coming in, and then there's a cell here, there's a little connection between them. And so this is, I always think, kind of like, just one of the most amazing things about the brain. Like, as far as we know, how our brains work is just by neurons sending out information in this form of these action potentials, and then they make synaptic connections. So this axon comes in and makes a synaptic connection here. This is then going to make a synaptic connection with the Purkinje cell. The Purkinje cell is going to send its axon and then make a synaptic connection outside of the cerebellar cortex. So that's sort of how, as far as we know, that's how neurons work. So it's actually like, this, when I say that this is like going to change the output of the cerebellar cortex, it literally is the output of the cerebellar cortex, because it's only the Purkinje cells firing that's going to be transmitted. And so this is sort of how we think neurons are working, as far as we can tell. Yeah, it is really cool, I think.
And then we have another question about, so you talked about mice in your presentation today. Are there other animals that can be used to model different types of ataxia?
Oh, that's a really good question. I think the answer is yes, and it kind of depends a little bit on like what kind of question you're asking. So I know of work by a colleague who works on fruit flies. So this seems like fruit fly. How can a fruit fly have ataxia? It doesn't necessarily, because it doesn't necessarily have the same sort of brain organization at all. Like, their brains are pretty different.
They're very small.
If you, yeah, exactly. But if you use, if they take a protein, this is a different kind of ataxia, and you put it into the fruit fly, you can see that there are some deficits, and that they can look then to see how the fruit fly, like, so not necessarily like, they don't have a cerebellum, they don't have Purkinje cells, but they do have cells, and the cells do misfire when they have the kind of protein that's looked for. What's the advantage of, why would you want to study something in a fruit fly? Well, the advantage is that you have, they're just really easy to use. They live only for a couple of days. And so you can kind of do these big screens where you could imagine that I'm going to take like my fruit fly that has a disease, and I'm going to do I'm going to put like 2,000 different genes in there mutated to see if any of them are modulating the disease gene. And that gives you kind of, when I say screen, it's like, it kind of gives you like new ways of thinking about the disease, because you're like, oh, wow, this seems to be interacting in a way that we didn't know yet. So it's a kind of very basic question, but it can lead us in new directions for like what might be interacting with this gene. So as far as I know, we don't have any disease models of like any fly models of SCA6. Sometimes people also use like zebrafish. So there are other animals, like kind of classic model systems that people use, and they often, it's just their advantages that they're easier to use and faster. Like mice are like, they're kind of
they take a lot of work, but they
take a
lot
of
results.
work, and we have to age them for 7 months before we can even use them. So that is actually kind of expensive. So yeah, I think they're great, but it depends on which question you're asking, for sure.
And with that, we are at time. So sorry for folks who we didn't get to your questions. But if you could join me each at your own computer screen, thanking Dr. Watt for joining us today and telling us all about SCA6. I hope you found this webinar helpful. And if you want to rewatch the recording, it's going to be on the National Ataxia Foundation website in about a week. Have a wonderful rest of your day, and thank you so much for joining us. Bye, everyone.
Thank you so much. Bye-bye.